Hello students, welcome you all to a new video of Sunanda's tutorial. In this video we will discuss about the transcription process in eukaryotes. Unlike prokaryotes, transcription in eukaryotes is a highly regulated, more complex biological process where specific segment of DNA is copied into mRNA or converted into mRNA molecule inside the nucleus of the cell. But in case of prokaryotes, the transcription as well as translation both takes place inside the cytoplasm because they lack nucleus. But eukaryotes have cell nucleus, so the transcription process takes place inside the nucleus of the cell. Okay. So there are three RNA polymerases which help in the process of transcription: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Okay, and the RNA polymerase 1 helps in the transcription of majority of ribosomal RNAs within the nucleolus. RNA polymerase 2 transcribes all protein coding genes into pre-messenger RNA and small regulatory RNAs. RNA polymerase 3 transcribes transfer RNA, 5S rRNA and other small structural nuclear RNAs. Okay. Next, the first step of this transcription process is the initiation. It is highly regulated stage and requires the formation of pre-initiation complex at the promoter site of the gene. Okay, so at first we have to recognize the promoter sequence. The core promoter features is an 80 rich region, so it should be 80 rich region. AATTT like this, or AAATT like this. Okay, so it should be 80 rich region known as Tata box, located roughly about 30 base pairs upstream of transcription start site. What is upstream? Where the transcription starts, or +1 site. Then transcription factors RNA polymerase 2 cannot bind directly to the DNAs. Instead, the transcription factors, okay, general transcription factors or GTFS, assemble step by step. So RNA polymerase actually helps in the process of transcription, but it cannot bind directly to the DNA or the main promoter sequence. So here the general transcription factors or GTFs plays the role. So TF2D via Tata binding protein or TPP binds directly to Tata box and bends the DNA or opens the DNA double helix. Then TF2A and TF2B attach to stabilize that complex and help to recruit RNA polymerase 2. Now the RNA polymerase 2 recruits here. TF2F accompanies RNA polymerase 2 to anchor it securely to the promoter region. Then TF2E and TF2H complete the assembly and form the initiation complex. Okay. Next, DNA melting takes place. TF2H acts as helicase enzyme and helps to open or unwind the DNA double helix to expose the template strand of the DNA. Then promoter clearance, TF2H also acts as kinase, which helps in the phosphorylation of C-terminal domain tail of RNA polymerase 2. Okay. This phosphorylation triggers the conformational change that releases the polymerase from the promoter so it can move down the gene. Okay. So this is how the initiation process takes place during transcription. Next, elongation. Once released, the RNA polymerase 2 moves along the template strand in 3 dash to 5 dash direction, synthesizing mRNA or complementary RNA in 5 dash to 3 dash direction. Okay, so mRNA, the newly synthesized mRNA have direction like this, 5 dash to 3 dash, and it is synthesized on the template strand which is in 3 dash to 5 dash direction. Then what is transcription bubble? Elongation occurs inside a safe localized loop of unwound DNA. Next, nucleosome management. Because eukaryotic DNA wound tightly bound to histones forming nucleosome, which is a specialized protein system called fat complex. It temporarily dismantles and reassembles nucleosomes ahead of and behind the moving polymerase. Okay, next the final step, which is termination. So it totally depends on the polymerase type, which polymerase helps in the process of transcription. For protein coding gene utilizing RNA polymerase II, termination is tightly coupled with some RNA processing. So at first, polyadenylation or cleavage signal, the polymerase transcribes past the actual end of the gene, copying specific polyadenylation signal sequence into nascent RNA. Unlike bacteria, there is no hairpin loop to stop the transcription. Polymerase II transcribes a specific polyadenylation signal sequence, which is the actual signal for terminating the transcription process. Next one is actual cleavage or RNA cut. So here specific cleavage factor. Okay, there are some cleavage factor like CPSF, CSTF. They recognize this signal sequence and cleanly cut the nascent pre-mRNA and free the polymerase chain. Okay. Next, the torpedo model. What is torpedo model? The remaining loose RNA still attached to polymerase is rapidly attacked by a fast 5' to 3' exonuclease enzyme. The enzyme degrades the residual RNA tail until it physically catches the polymerase, disrupting its grip and forcing it off the DNA template. So this is how the newly formed mRNA or pre-mRNA detach from the DNA template, and with this the whole process of transcription is completed. And with this I am concluding my lecture video on transcription of eukaryotes, transcription in eukaryotes. If you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's tutorial. Thank you.